Hello, very good afternoon to all of you. Good afternoon to one and all. We'll start in a couple of minutes. Yeah, good afternoon, Dhruvi. Good afternoon, Afreen. Good afternoon, all. So, friends, here in this session, we discuss. Yeah, Rupesh sir, good morning. Yeah, good afternoon. Sorry, good afternoon. So, friends, in this session, we discuss almost 10-15 years questions of the NEET examination. So, this discussion, the question discussion, is equally important for the students who is going to appear for the IIT-JAM, then JNU entrance examination, NEET examination. Sonu Sa, yeah, good afternoon. So the discussion would be very fundamental, and here we discuss approximately 50 questions from the last 15 years NEET examination. So some of the questions are very easy and some of the questions are a bit tricky. So which kind of care we are supposed to take, so we discuss here. So now let us start. So the first question, and along with me you can also write the answer in the chat box so that we can make some interactive session. So the first question, which of the following statement is not correct? See, this is last year's question. We discussed this year question in the last part of the discussion. So the question is, which of the following is not correct? So this kind of question, in this kind of question we should be very careful, right? Not correct, they are asking. So lysozymes are formed by the process of packaging in the endoplasmic reticulum. No, they are not synthesized and they are not formed in the endoplasmic reticulum. They are formed in the Golgi apparatus, right? So from the trans phase of the Golgi apparatus, we had discussion that, so if this is the Golgi apparatus, right? So this is the cis phase, right? This is the cis phase, forming phase, right? And then this is, we can suppose this is the trans phase. Okay, we can suppose trans phase. So from here the lysosomes, vacuoles, they bud off. Okay, lysosomes. So lysosomes are synthesized by or formed by the Golgi apparatus, not by the endoplasmic reticulum. So this choice is not correct, right? So now we come to the second choice. So lysosomes have numerous hydrolytic enzymes. So this is correct. So this seems correct, right? Third, hydrolytic enzymes of the lysosomes are active under acidic pH. Yeah, of course this is correct. Acidic pH has been maintained in the lysosomes by continuous pumping of the H plus inside the lysosome, right? And the fourth choice, lysosomes are membrane bound structure. Yeah, lysosomes are membrane bound structure. Okay, so the A number, according to the answer, A number is the correct choice, because they are asking not correct, isn't it? Yeah, A number. Afrin is correct. Abhay Kumar, yeah, you are correct. Dhruvi Patel, you are also correct. Let us discuss second question. So concept of omnis cellula e cellula. This is a very famous statement given by the Virchow, right? Rudolf Virchow. It is given by the Rudolf Virchow, and the cell theory after the addition of this statement, right? It was called modern cell theory, right? Modern cell theory, right? Cell theory was given by the Sliden and Swan, correct? But after Sliden and Swan, Rudolf Virchow, they added this statement, omnis cellula e cellulae. What does it mean? So it means that the cells comes from the pre-existing cells, or new cells comes from the pre-existing cells, right? So this statement was given by the Rudolf Virchow. So yeah, you all are correct. Vora, Afrin, Dhruvi, Abhay Kumar, yeah. Creative World, good afternoon, sir. Yes, good afternoon. Thank you. Yeah, Dhruvi Patel, cells are formed from the pre-existing cells. Yes, correct. So this is omnis cellula e cellula. See, the very easy questions asking in the NEET examination. These all are the NEET examination questions, right? Very fundamental question. Third question, which of the following statement regarding mitochondria is correct? Right? Mitochondria they are asking. So the mitochondrial matrix contains single circular DNA molecule, right? Yeah, this seems correct. Okay, single circular DNA molecule is there. See, this kind of question we should be very careful. So this is incorrect. They are asking incorrect, not the correct statement. Okay, so yeah, this is correct. Mitochondrial matrix, they are having single circular DNA they are having. Okay. Second choice, the outer membrane is permeable to monomers of the carbohydrate, fat and proteins. Yeah, outer membrane of the mitochondria is very much permeable, right? They are not selectively permeable, whereas the inner membrane is very, very selectively permeable, isn't it? The inner membrane of mitochondria is very selectively permeable. So outer membrane is not very selectively permeable, right? So this is also correct. B is also correct statement. A is also correct statement. Enzymes of electron transport are embedded in the outer membrane. No, this is wrong. Enzymes are embedded in the inner membrane of the mitochondria here, right? Enzymes for the electron transport. Alright. So the answer is C. Yeah, Dhruvi is correct. Yeah, Vaidehi, good afternoon. Yeah, good afternoon. Yeah, Abhay Kumar, you are correct. C. Yeah. Tisha, you are also correct. Yeah, according to the question, this is C statement, right? Fourth, we read here. So inner membrane is convoluted convoluted with foldings. Yeah, and they are called crista, right? They are called crista. Okay. So see, simple question. The necessity is that we should be careful, otherwise we can make mistake, because this is incorrect answer. So what happens during the examination, we are in very hurry actually. So we are just reading the first choice and we make the correct answer A. No, they are asking incorrect answers. Right? So the incorrect answer is C number. Okay. Yeah, you all are correct. Vora, Afrin, you are also correct. Good. Fourth number, the shorter arm and the longer arm of the submetacentric chromosome. So first of all, we should know this meaning of the submetacentric. So submetacentric means when the centromere is present above the center. Suppose this is the centromere, right? This is the centromere. So if the centromere is there, right, and this is the short arm, this is the long arm. So what does they call? They refer to as, right? This is called, the small arm is called P arm and the long arm is called Q arm. Remember it, right? P for, and this is, I think, French word, petite, right? Petite. Petite means short, right? Small p, they are writing, right? Small p and small q, right? So petite, they are short arm, okay? So the answer is P for the short arm and the Q for the long arm, right? This is, I think, not there in the NCERT, but this is asked in the NEET examination 2009, right? So keep remember it, right? So P is the short arm and the Q is the long arm, okay? Yeah. All right, Ajay Tiwari, yeah. Okay, all right. Okay. Next question. Which of the following pair of organelles does not contain DNA? See, many organelles, they contain DNA, we know, right? Many organelles like mitochondria, chloroplast, right? Mitochondria and chloroplast, they are having mitochondria and chloroplast, right? They are having their own DNA, right? They are having own DNA, and that DNA is circular, right? That DNA is not linear. Like bacteria, they are having the circular DNA, right? So now we come on to the question. So which of the following organelle does not contain DNA, right? So nuclear envelope and mitochondria. Yeah, nuclear envelope means they are talking about the nucleus, isn't it? So they have DNA. Mitochondria and lysosome. Here they are asking which of the following organelle does not contain DNA, right? So mitochondria, it contains DNA. Lysosome, they won't have DNA, okay? So this may be wrong. But chloroplast and vacuole, chloroplast, they are having DNA, but vacuoles, they don't have DNA, okay? Lysosome and vacuole. So here both choice, they don't have DNA, right? Here this contains DNA, this contains DNA, here both contains DNA, right? So according to the, see, what the student will do here, because they want to save the time, and in that consequence they make a mistake to answer B, right? Because they see, okay, yeah, mitochondria, they have DNA, but lysosome, they don't have DNA, so they just stick out the B and then they jump on to the next question. But they are making mistake, because the D number question is more suitable. You can see the lysosome as well as the vacuoles, they don't have DNA, isn't it? Yeah, so 5 number D, right? These, Trusab Baria, you are correct. Yeah, Ajay Tiwari, you are also correct. Alak, 5D. Yeah, 5D. So be careful, okay? 6 number question, matching. See, matching kind of question, what you do, you don't match, or you don't read this first of all. What you do, you just match in the question paper, right? So here we match. So Golgi apparatus, lysosome, vacuoles and ribosome. So this is the synthesis of protein. So ribosome, it synthesizes protein. On the surface of the ribosome, protein synthesis takes place, right? So factory of the protein synthesis. Then vacuole, they are considered as the dustbin, okay, or excess material inside the plant cell, they keep inside the vacuole, so trap waste and excretory product, right? Lysosome, they degrade the biomolecules and digesting the biomolecules, right? They are suicide bag, so this is D, right? And Golgi apparatus, so formation of glycoproteins and the glycolipids, yeah? So this is this one, right? Now you can match it, right? Yeah, 6B, Dhruvi is saying B. So A4, yeah, A4, B3, no, A3, right? A3, then B number is, A is the correct answer here, alright? A3, B4, then C2 and D1, okay? So this is the correct answer. Yeah, A, Kosa Patel is correct, A is the correct answer, very nice. Okay. Next question, which of the following is true for the nucleolus? They are asking about the nucleolus. So nucleolus is the organelle which is inside the nucleus, right? Suppose this is the cell membrane, this is the cell, and this is nucleus. So nucleolus is inside the nucleus, right? More than one nucleolus may be present, right? So this is the nucleolus, right? So which of the following is true? right? So first one, larger nucleoli are present in dividing cells. Not exactly actually. Dividing cells, they have larger nucleus, not the nucleolus. See, nucleolus is the site for the synthesis, or we can say the assembly of ribosome, right? Assembly, right? So this is the site for the assembly of ribosome. And see, ribosome is made up of two components, right? One is RNA, right? Okay, RNA, right? rRNA, and second is the protein, right? So what they do here in the nucleolus, the rRNA are there because it is inside the nucleus, so transcription takes place here, and they carry the protein from the cytoplasm, right? From the cytoplasm they bring the protein, and here they assemble on the nucleolus. So this is not correct. So it is a membrane bound structure. No, this is also one of the non-membranous organelle. Keep in the mind. Only 3-4 organelles are there which is non-membranous, like nucleolus, then centriole, right? Then ribosome. Ribosome is also without membrane, right? So these are the non-membranous organelle, right? Okay, so this is also wrong. So it takes part in the spindle formation. No, not at all. They are the site for the assembly of the ribosome, right? So it is the site for the active ribosomal RR. Yeah, this is correct answer. So this is the site for the active ribosomal RNA synthesis. Okay, RNA synthesis. Then only they can, they can be able to form the ribosome, active ribosome. So in the cells where they are actively synthesizing protein, right, they are having more amount of ribosomal RNA and nucleolus is big, right? All right. So D number is the correct answer. D, 7D. Yeah, Kosha is correct. Yeah, Dhruvi is correct. Yeah. Okay. Yeah, Ajay Tiwari. It's a collection of ribosomal RNA. Yeah, you are correct, Mr. Ajay. Yeah. Yeah, it's a collection of ribosomal RNA, right? Means rRNA, what we have written here, right? rRNA. Actually, one more information I'm giving here. There is a center which is called NOR, nuclear organizing region. This here a region, this region, nuclear region is called nuclear organizing region, right? So here the active rRNA synthesis is there. Okay, Mr. Ajay Tiwari. All right. Okay. Yeah. Now we move on to the another question, 8 number. So the 8 number, the Golgi complex participates in. Okay. So see, here we write ER as well as the Golgi, right? See, on the surface of the endoplasmic reticulum, right? On the surface of the endoplasmic reticulum, or we can say the rough endoplasmic reticulum, proteins we write RER, right? So protein synthesis is there, protein synthesis takes place, right? Then on the surface of the SER, lipid synthesis or related compounds are there, lipid. Then they are asking here Golgi complex. So on the surface of the Golgi complex, so see, the fatty acid breakdown is not there. So formation of the secret, yeah. First or second question we had discussion. The Golgi complex participates in formation of secretory vesicles like lysosome. Lysosome also is secretory vesicle actually, right? Then respiration in bacteria, no. Activation of the amino acid, not at all. So B number is the correct answer. Yeah. Trusa is correct. Vaidehi is correct. Dhruvi, yeah, correct. So formation of the secretory vesicle. So what happens? The RER or SER, when they synthesize protein, right? They synthesize lipid. They synthesize glycolipid here, right? So after that, they send those protein or glycolipids or lipids to the endoplasm Golgi apparatus, right? And then Golgi apparatus, the addressing takes place, right? Protein targeting takes place after that, right? And they comes out in the form of packed vesicle, right? And those vesicles are targeted to a particular organ. Okay, yeah, Afrin, you are correct. Ajay Tiwari, A and B. B, yeah, fatty acid breakdown. Yeah, you can say you are correct actually, but this is, if we have to select only one answer, so B is the most correct answer, right? So formation of the secretory vesicle. You are correct. Fatty acid breakdown some extent is there. I'm agreed. Okay. Yeah, correct. Next question. Which of the following event does not occur in the rough endoplasmic reticulum? RER, just we had discussion, right? So RER, which is not occurring on the RER, right? So rough endoplasmic reticulum, protein synthesis is there as well as folding is there, right? As well as folding also occurs on the surface of the rough endoplasmic reticulum or during the passage of from the rough endoplasmic reticulum to the Golgi apparatus. So in the meantime they fold actually, right? Then protein glycosylation, yeah, it also takes place. Protein glycosylation, that means after the protein synthesis, so lipid, I mean not lipid here, the carbohydrate residue is if supposed to be added, so they added onto the rough endoplasmic surface, reticulum surface only, right? Then cleavage of signal peptide, yes, cleavage of signal peptide is also there, right? So this is, you know, the protein targeting, right? What they are talking about, the protein targeting. So the signal for the protein targeting is cleaved here, right? Before the packaging into the, before it enters into the Golgi apparatus, right? So the signal peptide, right, was cleaved here. Fourth choice, phospholipid synthesis. No, phospholipid synthesis, they or it takes place onto the SER, right? So lipid related phenomenon, right? Lipid related phenomenon, whether it is phospholipid or whether it is glycolipid, so those phenomenon occurs on the SER, right? And they are talking about not occur on the, so not occur, so be careful, right? Not occur onto the rough endoplasmic reticulum, okay? Abhay Kumar, C is correct. Which question? 9, no, not C, D. D is the correct answer. See, a scientist called Blobel hypothesis, something which is called Blobel hypothesis. Maybe spelling would be wrong, this is name, right? Blobel was a scientist, right? Hypothesis. So he talked about the protein targeting. He received Nobel Prize also, Blobel, right? And if you read the Blobel hypothesis, then you find that signal peptide, they are made up of approximately, you know, 20 nucleotide, 20 amino acid long signal peptide. So signal peptide is cleaved into the, signal peptide is cleaved into the lumen of the rough endoplasmic reticulum. So this is correct. Okay, okay, 9 number. So select the correct match. Okay, this is very nice question. So see, something which is called giant chromosome, right? Giant chromosome. So chromosome. So two examples of giant chromosome here is written. So this is Lambrus chromosome and another is the polytene chromosome. These are the giant chromosome. First information we are learning from the question, right? So the Lambrus chromosome, incorrect match they are asking, no? So Lambrus chromosome, they are there in the they are there in the prophage 1 Lambrus chromosome. It present in the Lambrus, I am writing here LC only, right? Lambrus chromosome, they are present in the prophage 1, okay? Prophage 1, I mean prophage 1, one subphages is there, that is called diplotin, right? Diplotin, right? So in the diplotin stage it comes, right? And this is very, you know, big chromosome. What happens actually here, the active part, I mean the euchromatin, number of euchromatin is too much on this chromosome, right? So they are correctly matched here actually. So they appear in the diplotin stage and they appear as a bivalent. Allosome, so allosome, they are see, allosome, autosomes and allosomes. Chromosomes has been divided into two parts, autosome and allosome. So this is allosome and here they are matching with the sex chromosome. So they may be a pair, right? We should read the other choices also. Submetacentric chromosome, L shape, yeah, submetacentric chromosome will look like L. I mean, suppose this is the long arm, this is short, this is the centriole and centrosome, and this will be short arm. So this can look like L shape, right? So this is also correct. Then polytene chromosome, they are present in the oocyte of the amphibian. No, this is wrong actually. See, polytene chromosome, they are present in the salivary gland actually, right? Yeah, this is, I mean, they are present in the salivary gland of many insects actually. Okay, there is a class which is called dipterans in the insect, I mean in the arthropoda. So mostly they order, this is the order, so they found in the dipterans mostly, right? And their salivary gland, they are having the polytene chromosome, not in the oocyte. I mean this lambrous chromosome is present in the oocyte of the amphibians, right? So this lambrous chromosome is present in the oocyte of. So this is informative question actually. So this D would be the correct answer here. Okay, D. I mean not matched or incorrectly matched. Yeah, Ajay Tiwari, D, you are correct. Abhay Kumar, sir, this is not in, yeah, this is not in the NCERT textbook, right? This is not in the NCERT textbook. Yeah, you are correct. Ajay Tiwari, phospholipid synthesis occur in smooth endoplasmy. Yeah, you are correct. Phospholipid synthesis, correct. You are correct. Yeah. 9D. Yeah, all of you are correct. Very nice. Now, which of the following cell organelle is responsible for the extracting energy from the carbohydrate to form ATP? This is a very simple question, and quickly you can write the answer. D. Yeah, D. No. Okay, Ajay Tiwari. Okay, it was the earlier question. Okay, no. So which organelle ATP synthesis takes place? Abhay Kumar. No. You are talking about the previous question. I mean Ajay Tiwari was talking about the earlier question. Earlier it was the D answer. Yeah. So here, which organelle? Please write the answer. Mitochondria, isn't it? So mitochondria is the organelle which or where the ATP synthesis takes place. Yeah, Dhruvi Patel, C. Yeah, you are correct. Okay, Trusa, C. All of you are correct. Very nice. Good. This is a simple question actually. And these are the questions which you can solve within 2 to 3 seconds, right? So that you can save your time for solving the physics and chemistry questions, right? But don't solve question in hurry. बहुत हड़बड़ नहीं करना है, आराम से बनाना है, क्योंकि मार्क्स 4 ही हैं सबके लिए. Whether it is tough question or simple question, you are awarded with the 4 marks only. Okay? चाहे वो फिजिक्स के क्वेश्चन हो, केमिस्ट्री के क्वेश्चन है, बायोलॉजी के, all of them having the 4 marks. So select the mismatched 12 number. Gas vacuole, green bacteria. So mismatch, right? So be careful, right? So mismatch. Tell me the answer if you are able to solve. So gas vacuole, they are there in the green bacteria. Yeah, gas vacuoles are there in the green bacteria. Large central vacuole in the animal cells are present. Animal cells, do they have vacuole? No, not at all, right? So this may be mismatched, but we have to read the all 4 choices every time. Don't do that mistake. So not reading the choices, right? Protista is a kingdom which is the eukaryotes. I mean unicellular eukaryotes, right? Unicellular eukaryotes are kept into protistans. Okay, so this is correct. Then methanogens, they are prokaryotes. Yeah, methanogens, they are monerians actually, like archaebacteria, right? Archaebacteria, they are monerians, right? So they are prokaryotic. All right. Yeah, correct. 12B, Vaidehi is correct. Vorafrin is correct. Yeah. Others can also write your correct answer. Yeah, good. Yeah, next question. Now we move on to the next question. So next question is, see, you can see the level of the question almost, questions they are asking from the NCERT only. Very few questions are there from the out of NCERT, right? So 13 number, select the wrong statement, right? Wrong state. This is very good question actually. This is good question. 13 number. So select the wrong statement. So bacterial cell wall is made up of peptidoglycan. This is correct or wrong? This is correct. So bacterial cell wall is made up of peptidoglycan, right? Then pili and fimbriae. Pili, the small projections, the small hair-like projections on the surface are called pili. Okay, this is nothing but the extension of the cell membrane. So this is pili and fimbriae, right? Same. I mean pili, same. Almost similar structure is there. Are mainly involved for the motility of the bacterial cell. No. Fimbriae and pili, they are not meant for the motility. They are meant for the attachment. What they meant for? They are meant for the attachment to the substratum or attachment to the host tissue or cell. Right? So this seems wrong. C number, cyanobacteria lack flagellated cells. No, not at all. Then mycoplasma is wall-less microorganism. Yeah, no cell wall is there. Right? When bacterial cell wall is devoid, if you remove the bacterial cell wall, I mean the mycoplasma are the organisms which are not having they lack cell wall, right? So this is also correct. So the correct answer here is the B. Yeah, 13B. Yeah, you all are correct. Yeah, Shreyas Patel. Yeah, you are correct. You are wrong actually. Shreyas Patel. Yeah. Correct. You are correct. I'm sorry. B number. B is the correct answer. Yeah. Okay, very good. Then 14 number, all organelle containing hydrolytic enzyme is a cell organelle. A cell organelle contain hydrolytic enzyme. So this is very simple question. Lysosome, straightforward question. Lysosome, they contains hydrolytic enzyme. All right. Now we move to the next question. Mitochondria and chloroplast are. Right, they are asking mitochondria and chloroplast. So mitochondria and chloroplast are. So mitochondria and chloroplast are semi-autonomous organelle. This is correct, right? Because we had discussion that they are originated through a symbiotic relationship between the prokaryote and eukaryote during the course of evolution. So prokaryotic cell, they comes inside the eukaryotic cell. This is eukaryotic cell and this is the prokaryotic cell. This somehow comes inside and they resides there for forever, right? And they exist here in the symbiotic association. I mean both gets benefited, right? The bacteria here, I mean now we can call them the chloroplast. They got space and and in response to that they are giving the ATP, synthesizing ATP, which are essential for which are required for the eukaryotic cell. So first number is correct. The origin of the chloroplast is there in the same manner, we know. Then second is formed by division of pre-existing organelle and they contain DNA but lack protein synthesis machinery. See, they do have a protein synthesis machinery. Please listen carefully. They formed from the pre-existing organelle. Yeah, they divide by the fission, binary fission, chloroplast as well as the mitochondria. They contain DNA also. Yeah, they have DNA. This is also correct. But lack protein synthesis machinery. No, this statement is wrong here. They have the protein synthesis machinery. But see, carefully you listen, both chloroplast and mitochondria, they can synthesize only 5% protein of their use from their own genome, right? By own genome means what? The DNA they are having, circular DNA, only 5% protein they can synthesize. 95% protein in the mitochondria and chloroplast, they comes from the cytoplasm only, right? From the cytoplasm it comes actually, 95% protein in the mitochondria, right? So this is the information I am giving you, right? So be careful, note it down. So here the A is true, B is false. Yeah, this seems correct. Both A and B are false. No. Both A and B are correct. No, not at all. B is correct, but A is false. No. So correct answer is A. All right. This is good. I mean, some good informations are there in this question, right? So particularly this 5% protein, if they make the tough question, then they can, include this portion also. So how much percent of protein they can synthesize by their own genome? So only 5%, but at least they can synthesize. So that's it. And 95% they are exporting from the or importing from the cytoplasm. Okay. 16 number. So microtubules are the constituents of microtubules they are talking about. Microtubules are made up of. Okay. So microtubules are the constituent of. First number, centrioles and spindle fiber and chromatin. So yeah, centriole is made up of microtubule. Spindle fibers are also made up of, but chromatins are not made up of microtubules. Centrosome, nucleosome and centriole. Okay. See, the centrosome is the central part of the chromosome, or where the large and long and short arms are attached are called, that is centromere, right? That is centromere. Centrosome is the cytoplasm which is surrounding the centriole. Centriole is the perpendicular body near the nucleus. If this is a cell, this is centriole, and the special cytoplasm is surrounding the centriole, right? And that cytoplasm is called centrosome. Okay, all right. This cytoplasm is called centrosome. So centrosome is cytoplasm. So this is also wrong, right? Then cilia, flagella and peroxisome. Yeah, cilia and flagella is made up of the microtubule, but peroxisome is not made up of. Spindle fiber, centriole and cilia. Yeah, this is the correct answer. So all three here, spindle fiber, centriole and cilia, they are made up of microtubules. All right. So here you just keep in the mind the centrosome. Centrosome is the cytoplasm which is surrounding the centriole. This is the centriole, right? The perpendicular body is centriole, right? And when we talk about the chromosome, this is the long and short arm. So this is the centromere. These are the three confusing terminology actually, right? Centromere. Okay. All right. Yeah, microtubules are, yeah, you are also correct. You are correct, Mr. Tiwari. You are correct. Microtubules are eukaryotic. Yeah, eukaryotic cells they are having. Yeah, correct. All right. So prokaryotic flagella and prokaryotic cilia they don't have. Yeah, note it down. Okay. So prokaryotic system they don't have the microtubule in their cilia and flagella. Okay. 17. So which one of the following cell organelle is enclosed by a single membrane, right? So single membrane. This is very simple question. So lysosome is a single membrane system, right? Nuclei, this is double membrane. Mitochondria, double membrane. Chloroplast, double membrane. Okay. Outer and inner membrane is there. And in most of the cases where the two membranes are there, so outer membrane is more permeable, right, than that of the inner membrane. So correct answer is A. Okay, so A here is the correct answer. Yeah, Dhruvi, you are correct. Shreyas Patel, yes, correct. Okay, this is simple question. Vaidehi, A. Yeah. Now 18 number question. So water soluble pigments in plant cell vacuole are. This is important question, and 2-3 times they asked actually, right? If you solve 30-32 years questions, you will find 2-3 times this question has been asked. So can you answer me? 18 number. So 18 is the anthocyanin. Anthocyanin is the water soluble pigment, right? So this is the water soluble pigment, right? Anthocyanin, and it provides color to the flower, right? Color to flower, fruits, etc. Right? Yeah, 18A. Yeah, Mr. Tiwari, you are correct. No, not 18. 18A is not carotenoids. Yeah, no, carotenoid. In the vacuoles, they are talking about vacuoles, and in the vacuoles, anthocyanin is the pigment which is present, right? Yeah, Ajay Tiwari, yeah, B. Now you are correct. Yeah, anthocyanin. Okay. All right. And see, one more thing I want to add here, that this is also very important for you. The carotenoids, xanthophyll and chlorophyll. See, these are the pigment which are fat soluble, right? ये तीनों pigment क्या है? They are fat soluble और lipid soluble. Fat मत लिखो यहाँ पे, right? Lipid soluble क्या है ये? Lipid soluble. ये भी पूछता है क्वेश्चन कि which of the following is lipid soluble, which of the following is water soluble, right? So the anthocyanin is the water soluble pigment. Okay, all right. Okay, this is important question and good information. Okay, then 19 number, very long question it seems, but very simple question. Match the column and identify the correct option. Again, you just match first and then you try to fish out the answer, right? So thylakoid. So thylakoid is nothing but the flat membranous sacs in the stroma, right? In the thylakoid. Cristae or crista is nothing but the folding in the mitochondria, right? So this is the folding in the finger-like projections of the inner membrane of the mitochondria, right? Then Cristae, then cisternae, right? They are talking about cisternae. So cisternae are the component of the Golgi apparatus, right? Golgi, ये Golgi का part होता है ना. Cisternae, tubules and vesicles. These three components are there in the Golgi body. So cisternae. So this is the disc-shaped sac present in the Golgi apparatus. Chromatin, so condensed structure of the DNA. Okay, so A1, right? A का कौन सा अभी? A is 3, so A3, then B4, D number, yeah, D number, and D3. D का कौन सा? 2, D2, yeah, D number is correct. Yeah, Dhruvi, you are correct. Trusha, you are correct. Abhay, correct. No, Abhay, you are wrong actually. Abhay, no. Here matching is there. You just match it and fish out. So ये भी इसमें भी गलती होती है, right? So be cool, right? And then you try to fish out the. Yeah, Brinda, D. Yeah, you are correct, Brinda. All right. Now, 20 number question. Which of the following structure is not found in prokaryotic cell? So mesosomes are there. So mesosomes are the, suppose this is cell wall. If this is cell wall and this is cell membrane, so mesosome are the foldings of the cell membrane where the respiratory enzymes are filled. So this is the mesosome, right? ये क्या है? So this is present in the prokaryotes. Plasma membrane, yeah, they do have plasma membrane. Nuclear envelope, no. Ribosomes, yeah, they do have. 70S type, they do have. Right? So answer is C. Yeah, you are correct. Trusa Baria, good. Then 21. Cellular organelles with membranes are. Okay. So they are talking about cellular organelle with membrane. Right. So endoplasmic reticulum, we talked about non-membranous organelle in the earlier question. So 3 organelles are there. Which one? So the organelle ribosome, then centriole, nucleolus, these are the non-membranous organelle, right? So here they are talking about the membranous organelle. So endoplasmic reticulum, yes. Ribosome, no. Second choice, lysosome, yes. Golgi apparatus, yes. Mitochondria, yes. They are membranous organelle, right? So this seems correct. Nuclei, yes. Ribosome, no. Mitochondria, yes. Chromosome, they are talking about chromosome. So chromosome, no membrane is there. Ribosome, no membrane is there. Endoplasmic, yes. So correct answer is B. Yeah, good. Correct. Then 22, which of the following are not membrane bound? Again, same question they asked, right? See, in the 2015 only they are asking, almost same question, right? Which of the following is not a membrane bound? Okay, so now you tell me the answer. Not the membrane bound. So which one? Ribosome. This is very simple question. Yeah, 22D. Yeah, Abhay Kumar, correct. Brinda, you are also correct. Very good. So very simple question they are asking, right? DNA is not present in. So nucleus, DNA is present. Yeah. Mitochondria, they are having DNA, circular DNA. In mitochondria, they are having circular DNA. Okay. Chloroplast, yeah, they are also having circular DNA, double stranded circular DNA. Here also double stranded circular DNA. Right. Ribosomes, they don't have. They are having RNA. They are having rRNA and protein. Right. Ribosome is made up of rRNA plus protein, right? Protein plus rRNA. Okay, so this is correct answer, D number. So 23 is D. Yeah, Ajay Tiwari, you are correct. Yeah, Dhruvi, correct. Bora Afrin, you are also correct. Very nice. Okay, next question. Nuclear envelope is derivative of. This is good question. 24. Can you tell me the answer? Nuclear envelope. This is a good question. We can consider this a good question. See, microtubule, no. Microtubule, there is no such any contribution in the formation of membrane envelope or nuclear envelope. Yeah, inside the nuclear envelope, vimentine is there, and that vimentine is nothing but the microfilament, vimentine, right? Vimentine, you know, suppose this is cell and this is the nuclear membrane. These are the nuclear pores actually, right? Nuclear pores. So here vimentine layer is there, right? Vimentine layer is there inside to provide the support. Okay, so that vimentine is microfilament actually. That is not also microtubule. So nuclear envelope is not microtubule. Rough endoplasmic reticulum. Rough endoplasmic. See this, you know, rough endoplasmic reticulum is in continuation with the nuclear envelope, isn't it? Continuation में होता है nuclear envelope के continuation में, and the nuclear envelope is actually derived from the rough endoplasmic reticulum. Okay, nuclear envelope, right? Because proteins, too much of protein synthesis is there, and many more things are there. So it is made up of rough endoplasmic reticulum. So 24, D is the correct answer, not D. No, not D. Membrane of Golgi, no, not Golgi complex, not a smooth endoplasmic reticulum. See, see, I'm just sketching a cell, big cell, and this is the nucleus, right? And here you can see the outer membrane, and this is the, you know, this can be considered as the Golgi apparatus. And what happens actually here, it is in the continuation, right? Other Golgi, other rough endoplasmic reticulum can also be attached here. So what happens here, see, mostly in most of the cases you will see the ribosomes are present, or RER is present near to the nucleus, near to the nuclear surface, right? And smooth endoplasmic reticulum and rough endoplasmic reticulum, both are interconvertible actually, right? As per the requirement of the cell, right? So most of the time you will find the RER is present near the nucleus, right? So because RER, you know, the job of the RER is also to synthesize the nuclear membrane. That's why they are there with the nuclear membrane, or is in the continuation of the nuclear membrane. Sir, I say B. Yeah. Ajay Tiwari, you are correct. Then you are correct. Yeah, correct. Good. This is good question. Okay. Next. The structure that are formed by stacking of organized flattened membranous sacs in the chloroplast are called. This is again very simple question. Okay, can you tell me the answer? Stack of, see, stack of thylakoid. They are asking stack of thylakoid are called grana, right? They are called grana, right? Okay. The structure that are formed by stacking of the organized flattened membranous sac. So this is thylakoid. Okay, and a stack of thylakoids are called grana. Okay. Yeah, D number. By the D number. Yeah, you are correct. Yeah, Abhay Kumar, grana. You are also correct. Good. So see, my friends, very easy questions are there. So be careful and try to struggle with the question, right? Okay. 26th number. Select the correct matching in the following pairs. Correct matching. See, similar question asked 2-3 times earlier. Okay. So rough endoplasmic reticulum, synthesis of glycogen. What is glycogen? So glycogen is nothing but the polymer of glucose. Okay. This is the polymer of glucose. So rough endoplasmic, which of the following matching, select the correct matching pair. So glycogen is not synthesized on rough endoplasmic reticulum. What we learned in the earlier question, that proteins can be synthesized, right? Proteins, glycoproteins, right? Glycoproteins can be synthesized on the rough endoplasmic reticulum, but not glycogen only, right? So this is wrong, right? So this is wrong. Rough endoplasmic reticulum, oxidation of fatty acids. No, not at all, right? Fatty acid oxidation, it takes place on the smooth, degradation also on the smooth endoplasmic reticulum, bit extent Golgi apparatus also, right? Then smooth endoplasmic reticulum, the oxidation of phospholipid. No, they are normally synthesizing, not oxidizing, right? So fourth number is the smooth endoplasmic reticulum, then synthesis of lipids. Yeah, this is correct. Yeah. Yes, Shreyas Patel, you are correct. D number. Vaidehi also D. Okay. Yeah, all of you are correct. Good, very nice. Okay. Next question. The chromosome in which centromere is situated close to one end. See, this is very confusing question. Can you tell me the answer? One end. See, I am sketching here a chromosome. Suppose this is the centromere and this is short and long arm, right? This is one thing, right? And another is, this is one, right? And we can sketch here this also. So one is like this, right? So what they are asking, in which centromere is situated close to one end, close, not at the end, close to the one end. So this is the situation, right? They are talking about this one, isn't it? They are talking about this one. So this chromosome are called acrocentric. This is called telocentric, right? So they are asking close to one end, not at the end. If they ask about the end of the chromosome, if the centromere is there at the end of the chromosome, then it is telocentric. What they are asking? They are asking one close to the one end of the. So that's why this answer is acrocentric. Yeah. All of you are correct. No, Mr. Tiwari, you are not correct actually. 27. C metacentric means if it is at the center. If P is equals to Q, right? This is short arm or long arm. You cannot differentiate between the short and long arm, right? Then it is metacentric, right? So they are not talking about the metacentric. They are close to the one end. That's why acrocentric. Okay. Yeah, Dhruvi is correct. Okay. 28. Which of the following is not an inclusion body found in the prokaryotes? Yeah, inclusion body. So first of all, we should know the inclusion body. So inclusion body is the precipitated substance which is there in the excess, right? If something in the bacteria is there in the excess, right? if something there in the excess, then those substance has been precipitated to form a crystal like structure actually, right? And not exactly crystal, so those structures are called inclusion body, right? So which of the following is not an inclusion body? So see, first number choice, glycol, yeah, okay, yeah, Mr. Tiwari, yeah, correct. So 28. So glycogen granules, yeah, of course. If the excess glycogen is there, then they precipitate. See, the excess food cannot be reserved in any organelle in the prokaryotes because they don't have organelles. So what they do, they simply precipitate. Okay. So here the glycogen, they may precipitate, yeah, of course. Polysome, no, polysome is the structure which is the combination of mRNA and ribosome, right? So these are the ribosome, and this thread like structure is mRNA. So when the ribosomes are arranged, linearly arranged on the mRNA, then it is called polysome or polyribosome, right? They normally, this structure normally made during the protein synthesis, right? So when the mRNA has been synthesized, then the ribosome will bind, right? And they form polysome or polyribosome, right? Many times they asked this type of question, right? So polysome here is the not a exclusion body, right? So this may be the correct answer. Phosphate granule, yeah, they precipitate. 28B, polysome also, no, how? No, polysome is not the correct answer here. Yeah, Sinophycean granule, yeah, Sinophycean granule is the precipitated one. I mean, they are the inclusion body, right? No, Mr. Tiwari, I mean you mean polyribosome. No, polyribosome is not precipitated. Then how they synthesize the protein, right? They are not precipitated. They are floating in the cytosol actually. They are floating in the cytoplasm. Yeah, you deleted. Okay, right? I hope you are clear with this. Okay, yeah. So polysome is the complex of ribosome and mRNA. Many ribosomes are arranged linearly on the mRNA. Then it is polyribosome. All right. Yeah. Next question. The solid linear cytoskeletal element having a diameter of 6 nanometers. See, the cytoskeleton, LETON, cytoskeleton is divided into 3 types actually. 3 types में divide किया. कौन सा? तो microtubule मैं short में लिख रहा हूँ. Then microfilament and then intermediate filament. ये diameter के हिसाब से, right? They divided on the basis of diameter. So this is 25 nanometer approximately. This is approximately 10 to 15 nanometer, right? And this is approximately 5 to 8, you can say, right? Nanometer, right? So they are talking about 6 nanometer and made up of single type of monomers are known as, and singlet is there and single monomer is there. So the correct answer is I'm sorry, I'm very sorry. This is wrong actually. This is wrong. This is 5 to 8 nanometer. I'm very sorry. This is intermediate, means दोनों के बीच में ये होता है. So 5 to 8 nanometer and this is 10 to 15 nanometer. All right. I'm sorry. Okay. So the intermediate filament is 10 to 15. So this is microfilament. Okay. This is microfilament 5 to 8. 6 nanometer they are asking. So this is microfilament. All right. Yeah. 29B. B number is the correct answer. Abhay Kumar. Yeah. Yeah, Mr. Tiwari. Correct. So 30. The osmotic expansion of cell kept in water is chiefly regulated by, of course, the vacuole. This is a straightforward question, right? So excess water, it goes inside the vacuole, or if the water content is low, then it takes water from the vacuole only. So water balancing organelle is vacuole in the plants. All right. Next. Now match the following. Centriole, you just match it. So centriole, chlorophylls, cristae and ribosomes, right? So the cristae is the enfolding of the mitochondria, okay? Then the chlorophyll, they are there in the thylakoid membrane, right? Then cristae to ho gaya. Then ribosomes, they are okay, enfolding of the mitochondria, chlorophyll, thylakoid. Then ribozymes, I'm sorry, this is ribozymes, not ribosomes. Ribozymes are the RNA when acts as enzyme, right? RNA as an enzyme, right? Then we call them ribozyme, right? So ribozyme is nucleic acid, whether it is RNA or DNA, we can call them nucleic acid, right? And then centriole. So centriole and the basal bodies of the cilia and flagella, they are made up of same component, है ना? Microtubules से बने होते हैं. So you can now match 31 का 4 हुआ A का, then B is 2. Yeah, first number is the correct answer. Okay. Yeah, 30 का B. Okay. 31 is A. Correct? Yeah, Trusha. Correct. 32. The Golgi complex plays a major role in Golgi complex they are talking about. Okay. So the Golgi complex as energy transferring organelle, no, not at all. They won't transfer energy, right? In post translational modification, yeah, this seems correct, of the protein and glycosylation of lipids. Okay. The Golgi complex is responsible for the post-translational modification. Okay. So this seems correct. Third choice. In trapping the light and transforming into the chemical energy. No, this job will be carried out by the chloroplast. Fourth choice. In digesting proteins and carbohydrate. Not at all. No, they won't digest actually. So B number is the correct answer. So post-translational modification, they take place on the Golgi complex. So this is the major role actually, major role of the Golgi complexes. See, if we talk about the digesting proteins, so to some extent this is correct also. Why? So in the acrosome is the structure which is present on the sperm head, right? If this is sperm, right? So this is the acrosome, right? And in the acrosome, Golgi apparatus is there, rich in Golgi apparatus, right? And this Golgi apparatus synthesizes enzymes, right? That is called sperm lysine, right? So sperm lysine यहाँ पे कुछ हद तक ये सही है, लेकिन most major role, so major role is the B. Okay, sperm lysine is synthesized by the Golgi complex only, right? But here the answer is B, correct answer, right? Okay, good. Next, 33. Which one of the following organelle in figure is correctly matched with their function? So this organelle, it looks like rough endoplasmic reticulum because ribosomes are there. See, white dots are there. So ribosomes are there. Now you can search out the answer. So rough endoplasmic reticulum and protein synthesis is there on the rough endoplasmic reticulum, isn't it? So B number is the correct answer. 33B. Alright, this is not Golgi apparatus, so wrong. Rough endoplasmic reticulum and formation of glycoproteins, no. Golgi apparatus, no. This is endoplasmic reticulum, and endoplasmic में कौन सा? Rough endoplasmic reticulum. This is the major site of synthesis of lipid. Many times this question again comes, right? Smooth endoplasmic reticulum. Answer is smooth endoplasmic reticulum. So major site of the lipid synthesis is smooth endoplasmic reticulum. See, you can see how they are asking question, from where they are asking question. Almost similar question they ask many times. So you know, friends, when you solve 10, 15, 20 years question, you will automatically get good belief, actually good belief, and very good confidence level you will get, right? And apart from that, you will get the idea from where the questions they are asking, isn't it? Next, the term glycocalyx. This is very nice question actually. This is very nice question. Glycocalyx. See, glycocalyx, here we read the choice first, then we discuss. A layer present between the cell wall and the cell membrane. No. Cell wall and cell membrane in the bacteria. This is suppose cell wall. I am making bit thick cell wall, right? This is cell wall, and by other color I am making here, this. Just a minute. I am making here black color cell membrane, right? So cell membrane, these are the mesosome, right? Mesosome, right? And the space between the cell wall and cell membrane is called periplasmic space. This is called periplasmic space, right? This is periplasmic space, right? Okay, so here the cell wall, on the surface of the cell wall there is a slime layer, right? On the surface of the cell wall the slime layer is there, and those slime layers are made up of carbohydrate. Actually, suppose this green one is the slime layer, right? So this is called glycocalyx, right? This can be said as glycocalyx. ये इसको भी बोल सकते हैं glycocalyx. This is one thing. And another thing we learned in the cell membrane. Suppose this is cell membrane, right? I am making here. This is cell membrane, right? So what we studied in the cell membrane, the proteins are there. Suppose this is the proteins, right? And proteins are, they are having carbohydrate residue on the surface. This is also called glycocalyx. Now you read the choice and fish out the answer. So this is glycocalyx. See, function of glycocalyx here is the recognition, right? Like our blood group, ABO blood grouping, right? So they are due to the glycocalyx actually, right? So the glycocalyx here and here. Now you read the question. So a layer present between, no, this is wrong, right? Cell wall of bacteria, this may be correct, right? So note it down. So bacterial cell glyco-engineered to possess N-glycosylated proteins. Not at all. This is very, they are saying anything else, right? So this is wrong. So a layer surrounding the cell wall of the bacteria. So this is the correct question. Cell wall of bacteria, no. Cell wall of bacteria is made up of पेप्टिडोग्लाइकेन. सो बाहर की तरफ, ये जो हमने येलो कलर का यहाँ पे बनाया, सो बाहर की तरफ ऑन द आउटर सरफेस ऑफ द सेल वॉल. सो डी नंबर इज द मोस्ट करेक्ट चॉइस. ओके, ऑलराइट, दिस इज गुड क्वेश्चन एक्चुअली. और यदि यहाँ पे ये भी चॉइस दे देता तो ये बहुत ही अच्छा क्वेश्चन बनता है और कंफ्यूज होते बच्चे, राइट? दोनों जगह पे हम इसको ग्लाइकोकैलिक्स बोलते हैं. ओके, 36 नंबर. Which of the following type of plastid does not contain store food? See, three kind of plastid we have learned: chloroplast, chromoplast, and leucoplast. Okay, and here chromoplast is there, so color, ये color रखता है, they won't have any stored food material. Helioplast ये lipid store रखता है, they are the lipid storing, or I mean plastid. Aleuroplast protein, right? They store protein. And amyloplast they store carbohydrate, right? So not कह रहा है ना, which of the following is not contained? So answer is A number. I hope you got the point. Yes, Turusa, A. Chromoplast, it doesn't contain any stored food material. They normally contain pigments, and those pigments are required for the, they participate in the photosynthesis also, but not directly. They are antenna molecules, and they mostly impart the color of the fruit, flower, etc. Okay, right. 37 number question. So select the alternative giving correct identification and the function of the organelle A. Okay, so this is A. So this is mitochondria. Here we can see, right? So mitochondria produces cellular energy in the form of ATP. So this seems correct. Golgi apparatus, no. Lysosome, no. Endoplasm, no. This is very simple question. Okay. So this is the A. I mean, this one is the A. A is the correct answer. Okay. 38. Select the correct statement from the following regarding the cell membrane. Right. So sodium potassium ions are moved across the cell membrane by passive transport. No, this is active transport. See, if this is the cell membrane, right, and this is some protein through which the sodium potassium pump operate. See, we are making a complete cell, then it would be clear to you, right? If this is the cell and cell membrane, right, this is inside of the cell, this is inside, so cytoplasm, this is cytoplasm. So what happens in the sodium potassium pump? 3 sodium it will be pumped out, right? 3 sodium it will be pumped out, and in response to that 2 potassium it comes in. Here 2 potassium comes in the expense of 1 ATP. At the expense of 1 ATP it will carry this process. So sodium potassium pump is active transport, not the passive transport, because they use ATP here. Okay, so this is wrong. Second number, protein make up 60 to 70% of the cell membrane. This seems correct, but this is again confusing, because the protein content of the cell membrane, it depends actually. In many cell it is more than 70%, many cell less than 50% also, right? So keep in the mind, so read all the choices here. Third choice, lipids are arranged in bilayer with polar head towards the inner part. No, polar heads are towards the outside, isn't it? This is the polar head and they are situated outside. Okay, this is polar head and this is tail. Polar head are hydrophilic, right, and tails are hydrophobic. Alright, okay, so this is wrong. So fourth number, fluid mosaic model of the cell membrane was proposed by Singer and Nicholson. This is absolutely correct without any confusion. This is very good question, and normally I am telling you, during the exam or inside the exam hall, 90% student will make this question wrong, right? So this choice is also correct. I am not saying this choice is wrong. So this B number is wrong choice. This is also correct, but this is not correct for all kind of cell membrane. Okay, it vary actually. So D number is the most accurate answer. Yeah, Tiwari ji, plasma membrane contains 50 to 60% protein. Yeah, you are correct, but not always. Okay, Mr. Tiwari, not always. Like I'm telling you, some membrane they contain 60 to 70%, and even if you talk about the RBC, right, then they are having less than 50% protein, right? So it depends actually. Okay, so here the correct answer is D. Yeah. Next question. Yeah. Okay. What is true about the ribosome? Okay, so prokaryotic ribosome are 80S. No, not at all. They are 70S. So wrong. So these are composed of ribonucleic acid and protein. Yeah, this may be correct. RNA, rRNA, right, and protein is the constituent of ribosome. These are found in the eukaryotic cell. Yeah, this is all only only in eukaryote. So this is wrong. They are also present in eukaryote. These are self-replicating introns of some RNA. No, this is also wrong. So correct answer is B. B number is the correct answer. Now we quickly move on to the 40 number question. Okay. So 40 number, which of the following does not differ in E. coli and Chlamydomonas? E. coli is prokaryote, right? This is prokaryote, and Chlamydomonas is eukaryote, right? So ribosome, yeah, in both ribosome is there. E. coli they are having 70S ribosome, and eukaryotes and Chlamydomonas they are having 80S ribosome. So see, 80S ribosome in the cytoplasm, but they do have 70S ribosome also in the mitochondria and chloroplast, right? Mitochondria and chloroplast both are there in the Chlamydomonas. So Chlamydomonas they are having 70 as well as the 80S ribosome. So ribosome is there in both prokaryote and eukaryote. Chromosomal organization, no, this is not there in the E. coli, right? It is there in the Chlamydomonas because Chlamydomonas is the eukaryote. Cell wall is there in the E. coli. This is correct. Cell wall is there in the E. coli Okay, that is made up of peptidoglycan, right? See, the question is one of the following does not differ, right? Ribosomes they are having, but one is having only 70s, another is having 80 and 70 both has. Chromosomal organism not there. Cell wall, it is made up of peptidoglycan in prokaryote, right? This is peptidoglycan, and here this is made up of cellulose, right? Cellulosic cell wall is there. So this is also different. Plasma membrane is same, whether it is prokaryote, yeah, Dhruvi D, yeah, you are correct. So plasma membrane is correct in both. I mean, whether it is prokaryote or eukaryote, it won't matter. It remains same. Plasma membrane remains same in the prokaryote as well as in the eukaryote. Okay. All right. Okay. Next. Now we move on to the next. Which one of the following cellular part is correctly described? Okay. The choice is thylakoid, flattened membranous sacs forming the grana of the chloroplast. This seems correct. Centriole, site of the active RNA synthesis, not at all. This is nucleolus. Ribosomes, those on chloroplast are 80s, no, bilkul nahi. And cytoplasm 70s, no, wrong. Lysosomes, optimally active at a pH of 8.5, no. pH would be 3 to 4, acidic pH isko chahiye, right? So this is also wrong. So the correct answer is A. A number is the correct answer. Dhruvi, sorry, it's B. Which one? 40. Okay, all right. So this is A. Here the 41, the correct answer is A. So 42. Now we move on to the another question. Which one of the following structures is an organelle with organelle? Organelle within the organelle, right? Organelle within the organelle. Like you can see the mitochondria. If we talk about the mitochondria, so mitochondria is the organelle which is or which contains many ribosomes, 70s ribosomes. So ribosomes, this is the organelle within the organelle, right? So ribosome is the correct answer. Okay. Organelle within the organelle is the ribosome. Okay, all right. अब है 42 का C, नहीं, बिल्कुल नहीं. 41 A, yeah, 41 is A. 42 is not C actually. Organelle within the organelle is the ribosome, right? Ribosome is there inside the organelle. That's why this is called organelle within organelle. ठीक है? Next, the peptide synthesis inside the cell takes place in peptide synthesis. Ribosome, protein synthesis they are talking about. So protein synthesis is there on the ribosome, right? Protein synthesis factory hai ye, right? Protein synthesis is there on the ribosome. Okay. Then next question, important site for the formation of glycoproteins. Important site for the formation of glycoproteins and glycolipid is. Yeah, you can tell me the answer. Many times we discussed. Golgi apparatus, right? Glycoproteins and glycolipids are Golgi apparatus. Okay? Correct? Okay. Now 45 number. Which one of the following is not considered as part of endomembrane system? Yeah, endomembrane system. So endomembrane system, the endoplasmic reticulum will come, SCR, RER, Golgi apparatus will come, lysosome will come, vacuole will come, because lysosome, vacuole, it comes from the Golgi apparatus. So Golgi complex is there, vacuole is there, lysosome is there, peroxisome is not there. So 45 का आंसर हो जाएगा B, peroxisome. Okay. Okay, 43 is D, Dhruvi, Abhay Kumar Golgiya. Okay, all right. Okay. Then 46. The figure below shows the structure of mitochondrion with its 4 parts labeled A, B, C and D. Select the part correctly matched with the function. Okay. So D here it seems the outer membrane, right? So D is the outer membrane, B is the inner membrane, right? And A A here is the, it is not visible clearly, but C here is the crista, and B here I think this is the inner membrane. B, they are talking about inner membrane. This B is the inner membrane, right? So question is, the mitochondria with 4 A, B, C, D, select the correctly matched with their function. Okay, all right. So D is the outer layer and gives rise to the inner membrane folding. No. B, inner membrane forms the infolding called crista. Yeah, this is correct. C is the crista and possesses single circular. No. DNA, no. This is there in the matrix, mitochondrial matrix. And D is the, A is the matrix. Okay. And major site of respiratory, no. The major site for the respiratory enzymes are the inner membrane, right? Enzymes for the electron transport are situated in the inner membrane, right? So inner membrane, they contains many enzymes. So B is the correct answer here, right? Okay. Next question, the plasma membrane consists mainly of 47. Plasma membrane, it consists mainly of, yeah, 46B, Dhruvi, you are correct. Yeah, all of you are correct. So here the plasma membrane is proteins embedded in the phospholipid bilayer, isn't it? This number, proteins embedded in the phospholipid bilayer. Okay, so this is the correct answer. The first one is a phospholipid embedded in protein. No, not at all. Right. Proteins embedded in a polymer of glucose. No, not at all. Proteins embedded in the carbohydrate. No. So B number is the correct answer. Okay, you can see. Very easy. 48. The main area of various type of activities of cell is, simple question, cytoplasm. So all chemical reactions, not all actually, major or majority of chemical reaction takes place in the cytoplasm, right? Majority of chemical reaction, they take place in the cytoplasm. Okay, cytoplasm is the correct answer. 48, cytoplasm. Abhay Kumar, yeah, you are correct, Abhay. Yeah. (49) Which one of the following has its own DNA? Again, you tell me. This is simple question. Own DNA. Yeah. 47B. Yeah, correct. 47. This is 49. Own DNA. So mitochondria, they are having their own DNA, and that DNA is circular. Keep in the mind, circular, self-replicating also, right? Circular, self-replicating. Small DNA is there, but see, these DNA only synthesize 5% of the protein for the mitochondria. Right? ऐसा नहीं कि माइटोकॉन्ड्रिया में डीएनए है ही तो उसको जीनोमिक डीएनए से कोई मतलब नहीं है. They need 95% of the protein from the genomic DNA only, right? Okay, मतलब वो साइटोप्लाज्म से उसको चाहिए 95% प्रोटीन. ठीक है, या 48 is C. Okay, 50, last question we are solving now. Almost our time is over. So which one of the following structures between two adjacent cells is an effective transport pathway? Okay, so see, here they are not talking about the plants or animals, right? But one of the following structure between the two adjacent cells as an effective transport pathway, right? So in the plant, that cell-cell communication or cell-cell junction is called plasmodesmata, right? They are called plasmodesmata, right? Plastoquinone, no, they are the protein involved in the photosynthesis or photosynthetic pathway we discussed in the photosynthesis chapter. Endoplasmic reticulum, not at all, right? Plasma lemma, no, not at all. So plasmodesmata is there in the plants. Desmosome is there in the plant, through which they can communicate, right, from the neighboring cells, right? So here the plasmodesmata is the correct answer. Okay. Okay, so friends, here we discuss 50 questions and almost 10 years question we covered from the cell chapter. And here we can see most of the question they are asking is repetition of the other question, actually earlier question. And here you at least you noted down that till 2000 or 2000 to 2020 only questions they are asking from the NCERT. Very few questions we found. I think one question only, that was the giant chromosome, right? Giant chromosome, that was not there in the NCERT book. Okay, so actually it was there in the you know earlier version of the NCERT. It was there, but right now it is not there. But they asked in the 2018, right? Otherwise, normally 99% questions they are asking from the NCERT. So again, I suggest you go through the NCERT very thoroughly. Okay, all questions they are asking. Yes, okay. Even in the NET examination also, NET, GATE, SLATE, you should have very good command on the NCERT. Even the MSc students, they are reading the NCERT. Otherwise, they won't clear the NET examination, GATE examination, right? So the NCERT is very basic book, and if you are there in the 11th or 12th, so it is very good time to read, right? So friends, so here we end up our session, and again I'm thanking you to all the participants who make this lecture successful. And almost one day, after 1 day gap, I'm going to arrange this kind of activity for the different chapters. Live session would be there. So if you feel this lecture is useful for you, so you tell your friend, you can communicate to your friend, and you can forward this video to your friends, your colleagues, right? So that maximum can be get benefited from these kind of activities. Okay, so I hope to see you in the next lecture. Next lecture would be on Sunday, right? Sunday 4 o'clock to 5:30. So this is the fixed time where we meet. Okay, so thank you again, once again, and we'll see you on next lecture on Sunday. So thank you very much.